This I'm asking because the data is not published at the patent application file. So we looked at 5 patients. They have gone through at least 4 different type of treatments and still the cancer came back. So we're curious to see कैसा हुआ क्या, why, what is makeup of their उस biopsy के कारण कि 4 different type की treatment उससे हो चुकी है but it comes back. So we did, now you see analysis, we did micro RNA analysis looking at the micro RNA level पे कि कौन कौन से signaling pathways, कौन से proteins जो हैं वो over expressed हैं. So you see another technique that we use. I showed you about synthesis. हमने किया, हमने biopsy ली. I'm using the terms you hopefully now you can connect जो पिछले 4 दिन से बता रहा हूँ. We took the patient samples, did the analysis. They have gone through 4 different types of treatment and this is what happened. So what we observed was that there are pathways which are associated with microRNA processing and stability of the protein. वो जो pathways हैं, those pathways are significantly elevated in those patients. So you simply देखो कि उनके अंदर वो जो pathway हैं, अब there are thousands of those. उनको जब micro analysis करते हैं तो देखिए कौन से pathway, which pathways are before and after treatment. So we identified those pathways. Okay, those pathways are there. And so this is again 5 patient tumor samples associated with this. And then we looked at various different pathways. One of the protein that stood out as a pattern, जो I've been saying AI and pattern, machine learning and pattern, this is real data. और उसमें देखा कि one of the protein, processing protein जो उन pathways के अंदर काम करता है, इसे Y-box protein 1. It's a cold shock protein. It functions DNA associated also with RNA. So it's a sort of a transcription factor. So my terms which I'm using, if you do not follow, try to read them. It's a transcription factor. जो transcription होता है ना, जो आपको फिर से S phase, G1 phase, M phase, G2, वो transcript जो है आपका DNA की जो information है, RNA उसमें जाके transfer करते हैं नए cell के बनाने में, जो transcription part है. So there are many proteins that play. One of the protein is Y-box protein 1. So it interacts with DNA, it interacts with RNA. That's what its roles is, and many functional जो हमारे cell के अंदर, tissue के अंदर आपके body की functions है, it plays an important role. Now it is, for example, a few that I mentioned. It's a key regulator of RNA processing. Multifunctional cold shock protein regulates DNA and RNA in the cellular events. Cell के जो events हैं, I have listed here, for example. And now once they splice, transcription होता है उसकी packaging में, किस तरह से वो package होते हैं, नया cell बनता है, उन सब के अंदर Y-box protein plays an important role. And here is, so this is my PhD student who put together this whole, looked at the entire literature to see Y-box का role कहाँ है और क्या उसके नीचे ऊपर दाएं बाएं, what are the different pathways which are associated with it and how it gets influenced. So these are just simplified version. So it regulates within the cell many functions, and here the EMT जो है metastasis, जो सेल से बाहर निकल के फैलता है, मेटास्टाइज होता है, उसको फंक्शन को, apoptosis, killing the cell, right? ड्रग efflux पंप जो मैंने बताया उसमें भी उसका रोल है. इट जो स्ट्रेस होता है, जब बॉडी के ऊपर कोई स्ट्रेस, सेल पे जो स्ट्रेस होता है उसके रिस्पांस में इम्यून सिस्टम के अंदर इट हेल्प्स टू फंक्शन. सो अगेन सेल साइकिल के कंट्रोल करने में उसका फंक्शन है. सो दीज आर some of the factor and signaling pathways. So one single protein, inside the cell, it has so many different functions. This one protein, just imagine that protein is connected to another few protein, large protein, I don't know how many small protein. Each one has its roles to play within the cell, within the tissue, within the organ. वो सब जब बन के तैयार होता है, ये सुंदर रचनाएं पैदा होती हैं. It's a very complex system, right? So we started to look at के अगर ऐसा है, and there are various, these whatever EGFR, IGFR, ये जो different pathways या signaling हमें पता है, like ये as best as we know tumor microenvironment factors क्या क्या हैं, उसका drug efflux क्या होता है, EMT, इसके जो known proteins हैं, those who contribute, those are, as you see here, directly or indirectly are affected. So they are upstream to this, some of they are downstream to this protein. So we thought, okay, there is probably an opportunity. Okay, we start understanding this protein and try to design molecules which may interfere in the function of this protein in the cancer cells, so that cancer cell का जो है growth को कर दे. Now, before we started, we said, okay, let's first look in the literature and see what do we know, what is reported in the literature. So what I'm showing here is we looked at two databases, and what it shows is this is collection of patient, ovarian cancer patient, उनका YBOX1 protein expression, and 1, 2, 3, 4 here are the stages, and stage 1 cancer, stage 2 cancer, इस जो stages हैं, उन हर stage पे YBOX protein उनका expression क्या है. So as you see here, as it advances, YBOX protein becomes more and more active, even though after the treatment, which means it acquires resistance. So this is an acquired resistance. The more treatment होती जा रही है, वो और भी aggressive होता जा रहा है. That is one form. So and we looked at another database. ये the cancer science Singapore का है. Again, as you see, as the growth, the stages उसकी बढ़ती हैं, उसका expression बढ़ता जाता है even after treatment. So which means treatment से कोई फर्क नहीं पड़ता. Protein is something. We try to look at now. Okay, what are the proteins? Now ये तो ठीक है, YBOX protein is a complex. What are the proteins which go up and down when you after the treatment? So we identify few of these. Why CD44 is one molecule. MYC, c-MYC is a master regulator, one of the master regulator. There are many master regulators: P53, c-MYC, YBOX, snail, PIM1. They are kinases. We try to identify कौन से ऐसे हैं जो कुछ समझ में आए, so that you start preparing and start working around that. So and then we look at again okay, if YBOX1 is so important, so maybe there is an opportunity that you start developing a drug for targeting YBOX1, because अगर उसको आपने control कर दिया तो बाकी की जो उसकी जो functions हैं which influences the cancer growth, उसको रोक देंगे. So before that we again looked at the patient data to see कि क्या हो रहा है, and it clearly showed कि patients where there is a low expression of YBOX, they are responding better to the treatment compared to where there were a higher expression of YBOX1, which correlated with our hypothesis theory that targeting YBOX1 could be a way to develop a drug for this. Now go back and think to my first and second and third lecture. Okay, you have to first identify the target आपका क्या है. So we looked at the cells. We went through all the data that we could from the public domain, from the patient data or research data, and tried to make sense of. We did the analysis of the patient samples, try to understand at the microRNA level what is overexpressed in those patients, go back to patient sample data, उसको correlate करने के लिए कि really it is making sense या हमारा भ्रम है या there is something else to this. So when that correlation, all of that happened, now that gives you confidence कि ठीक है, we are on probably ठीक track पे हैं करने के लिए, and that's how piece by piece we put the information together. So now this is to correlate कि वहाँ तक पहुँचने के बाद में okay, from the patient data it shows कि जिनका high expression है, they respond poorly, which means this is a potential target जिसके लिए आप drug develop कर सकते हो. Simultaneously, my lab has been interested from the chemistry point of view. As a chemist, we've been interested to see drugs when they went to clinical trial, phase 1, phase 2, and they failed. They didn't go to market. तो उसमें क्या change था? What was wrong with their structure? Or so we were trying to, again, as I've been telling you guys, from the lens of chemistry, इसके structure में ऐसी क्या issue है, क्या problem है, that we can fix, so it becomes effective. So we started to look at those drugs. One of those drugs was etoposide. Etoposide is a natural product. It is a compound These are podophylotoxins. If you look at podophylotoxins are class, so this is a drug which is obtained from there are plants वो ठंडी जगह पे होते हैं. यहाँ पे फुटिला ऑफ हिमालयस नॉर्थ ईस्ट इंडिया में होते हैं. इट्स अबाउट अ मीटर हाई प्लांट जो है, एंड उसके जो उस प्लांट में से पोडोफाइलोटॉक्सिन्स होते हैं. और या फिर नॉर्थ ईस्ट ऑफ यूएस कनाडा जहाँ पे बहुत ठंडा वेदर होता है, वहाँ पे लगभग 1 मीटर जितने और 3-3.5 फीट के होता है ये प्लांट. उसके जो है डिफरेंट पार्ट्स में से ये कंपाउंड्स आते हैं. तो उसमें से इटोपोसाइड निकला था 80s के अंदर. As I told you, the natural product chemists are extracting and then they are testing. Etoposide turned out to be one of the compound which was effective. But for whatever reason, initially it was withdrawn. So, but showed toxicity and again it came back with uske analogs bane and it started to come back. So we were interested कि ठीक है उसकी toxicity remove करने के लिए as a chemist what is it that we can do. So this is basically a general structure which has 4 rings, and then ये X, R1, R2, all these substitution. We as a chemist started to design our own variation to see कि उसकी toxicity जो है उसको. Now toxicity उसका the reason was this right side. So when it breaks metabolite, okay, when etoposide goes inside it metabolites, enzyme उसको break करते हैं. One of the product you obtained is catechol. आपने catechol का नाम सुना है? It's a simple chemical. It's acidic. So सीधा जब जाता है तो साइड इफेक्ट उसका होता था अल्सराइटिस, अल्सर होने का, क्योंकि इसे हाईली एसिडिक कंपाउंड. सो वी थॉट ओके ठीक है, अब इसके जो कैटेकॉल की प्रॉपर्टीज जो होती है उस मॉलिक्यूल के रिंग का स्ट्रक्चर थोड़ा चेंज करते हैं. सो वो अंदर जाके बनेगा, वो कैटेकॉल नहीं बनेगा, इट वॉन्ट बी एसिडिक. सो दिस इज हाउ स्लोली स्टेप बाय स्टेप वी स्टार्टेड टू लुक एट कि किस तरह से हम उसको चेंज कर सकते हैं, किस तरह से हम उन चीज़ों को चेंज कर सकते हैं. वी स्टार्टेड टू डू दैट and try to come up with our own structure, newer structures. We did that and then made compounds, and we tested on 60 different cancer cell lines, 9 different tumor types: head and neck, renal, breast cancer, leukemia, ovarian, prostate cancer, colon cancer. So 9 different types we tested, and these are the the cell lines which are mentioned here. And it turned out this was separately happening. We didn't go for this project. We were separately as a chemist interested to see why drugs were toxic and not working, and we designed that. And we designed compounds which turned out to be highly effective against the ovarian cell lines. So we said, okay, from that project, from the chemistry studies, we identified, we designed a compound which is effective. Here we have shown in the ovarian cancer models that there is a protein that doesn't work. What if we start using this against these cell lines, ovarian cell lines, these compounds which we have developed, we identified, and we started testing the research part. So we are researching. Long story short, we identified the compound which were highly effective, and then we started to do in vivo studies. Took into the mice, implanted cancer cell lines, ovarian cancer cell line, did that, and you see this, the colors that you see, we did imaging to see where the tumor was. So how many things I have told you so far जो मैं पिछले 4 दिन में कहता आ रहा हूँ. Chemistry, microRNA analysis, assay development, right? Imaging techniques that we used, right? So we did all of these things, and these are the cell lines that we developed models against which we started testing these compounds. And you see ये जो है control which is without control which was used as a reference, असर कुछ भी नहीं होता. When we give our drugs, you see the tumor is suppressed. It is suppressed further. So we did these studies, mice 42 days of the treatment. Now, these words here, 10% PEG 300 in Insulin formulation. We prepared the formulation, सिर्फ DMSO में नहीं. Formulation that is not toxic to the cell, formulation which is tolerable by the cell, but formulation in which your compound is soluble. Formulation chemist. That and making it bioavailable. Because if formulation, whether it will take it to the site of action. If it doesn't, it's not going to be helpful. So you see how many of different expertise जो मैंने आपको बताया पिछले 4 दिन में मैं कहता आ रहा हूँ. And then this is before treatment with the vehicle, after treatment, tumor shrink. With another compound, and we use 20 mg per kg, right? And you see with and the second compound even further, it's suppressed. So then we did the pharmacokinetic studies. कि कंपाउंड देते हैं तो अपने ऑर्गन साइट तक पहुंचने में कितना टाइम लगता है, उसका हाफ लाइफ क्या है. You see, I'm using the term which I've been telling you for last 4 days. कि ड्रग का क्लियरेंस का हाफ लाइफ क्या, because it has to reach to the site. And we did that. You see here, liver जो है, these are again PK analysis, जो C max जो है 1 point, जो maximum concentration after you give the treatment कितनी होती है उस ड्रग की. So you can do again chromatography, do the analysis, which tells you can get up to 1.6 microgram per ml. Then you have to study and see, is this enough to get the effect that you need. So you, all of these Tmax, how much time, so highest concentration you get, what time it takes to get to that concentration. 5 मिनट के अंदर ही उसका असर आया था. So drug is effective. You give it and 5 minutes के अंदर उसका max concentration प्लाज्मा में निकल आती है. T half कितनी देर तक उसमें रहती है, right? All the things that I've been telling you. So we studied that. We studied that to find out कि कितनी रहता है. And then drug distribution, कि drug reaches to कौन-कौन सी sites में. So then these are the standard. You look at the organs, especially liver, ovaries में, क्योंकि because it was for ovarian cancer. Liver में, क्योंकि liver के अंदर liver metabolites इसको chop करते हैं, right? So you find out that concentration. And then toxicity. AST, ALT, these are toxicity for liver, और जो है आपका कि कितना वो toxic है. And basically what you do is you calculate the enzymes. वो enzymes से you can quantify कि उसका enzyme level क्या है, उसकी toxicity क्या होगी. So we did all of this liver toxicity for again 42 days. These and look for the enzyme. And this is after many years of work. We published this in this 2021. When we published, soon after that, around that time, a group in Harvard was studying this. They saw immediately in उनका जो reference, जब उनका पेपर रिव्यू के लिए आया, उन्होंने फिर से हमारा पेपर लेके उसमें डाला कि अब तक सिर्फ यही ड्रग है जो काम करता है. There is nothing available, because they were also independently studying Ybox1, and this is the only drug which is reported that works. So this is just to give you an appreciation of, from chemistry to assay development to model development, and you see how many terminology that I used. All of these are done to identify the candidate and then do all the PK studies. This is again ADME PK studies जो हम कर रहे हैं. And then we started to look at, okay, now this candidate, let's go further and try to see what it does. So we expanded that to see that it works in ovarian cancer. Ybox protein 1 is expressed, and I showed you those chart, patient chart as with the stages, overexpression बढ़ जाता है. But then see कि Ybox protein 1 है, और दूसरे कौन से कैंसर टाइप हैं जिसमें वो प्रोटीन का फंक्शन है. हो सकता है ये ड्रग उसके लिए भी काम करे, whether it's a breast cancer or head and neck cancer, lung cancer, जो भी है. So we started to look at all consequences. तो हमारे in my lab we're doing a lot of work on triple negative breast cancer. So we had models available. We said, okay, let's now look at triple negative breast cancer. We looked at that and we identified that there were actually even TNBC, triple negative breast cancer, also Ybox protein is highly expressed. So we said, okay, we have molecule, we have models, now let's start testing that. So this is the in vitro study that we did, various specifically breast cancer cell lines बनाई, models बनाए, उनके अंदर test किया. When it worked out, we went ahead and then created the various stains, blacks, 6 mice, लेके उनके ऊपर we did various these studies again to show 93, 92 जो 2 compounds है किस तरह से काम करते हैं. And as you see यहाँ पे you probably may not be able to see ये metastasis you can control. So sorry आपको नज़र नहीं आ रहे होंगे because of there are these arrows which actually show कि उसका effect क्या है in control condition पे. So metastasis is when it moves from one place to another place, and you can follow the enzymes. Ki67 is one of the enzymes. You follow that and it can tell you क्या Ki67 बढ़ रहा है या कम हो रहा है. You can see before and after treatment. You can tell whether your compound is working or not. So we took this, we tested in the lung metastasis studies के अंदर. So this compound was effective. This was the picture may not show, but look at the quantification. Vehicle के अंदर really doesn't work. बहुत metastasis होती है कि ड्रग जो कैंसर सेल हैं वो escape करके दूसरी यहाँ पहुँच जाते हैं और बढ़ाते हैं. When we gave our one candidate, it suppressed metastasis. When we gave the another candidate, it suppressed further, which means the drug on attacking Y box protein 1 was keeping it controlled locally rather than spreading. So you do these kind of study, and then we did a lot of studies to establish that your drug target is actually Y box protein 1. So there are many different assays that you could use. So this is an assay which is called SETSA. जो है the circular the thermal analysis, which is basically what is done is you take your protein of interest, and then you take the compound, you take a group of proteins and put the compound, and then you turn the stability of the protein. And if the protein, whichever protein जिसके साथ में आपका ड्रग इंटरैक्ट करता है वो प्रोटीन को पकड़ के रखेगा, तो उसका जो थर्मल शिफ्ट है वो रुक जाएगा, कम होगा. So you look for the proteins and then say okay, my drug or this drug इस प्रोटीन के ऊपर सबसे ज़्यादा शो करता है, which means that's the interaction mode. So you identify which proteins are affected by your drug. So that's called SETSA analysis. We did that, and we established that Y box protein 1 is actually the target. We did many, again, proteomic studies, genomic, whole cell genomics, proteomic studies to identify कि even with 2 mg per kg, how with the time dose dependence, कितने टाइम में उसका असर होता है, कितने डोज पे उसका असर होता है. So then you actually now is the development part. Once you have the discovery part, now is the development part to see how the drug can be developed. And this is all you see the proteomics to establish that the drug is working against that protein that you intended it to work on. Now, as I showed you, our interest was कि paclitaxel. My story started from why paclitaxel के अंदर resistance है. So research तो कर दिया. We started with the hypothesis कि Y box protein जो है is the culprit जो टैक्सॉल की जो है एक्टिविटी को लो करता है. अब अगर हमने नया ड्रग बनाया है तो भी उसको प्रूव करना पड़ेगा ना कि वाकई उसको रोकता है. Otherwise just a theoretical part. So what we did was, as you see here, we studied, these are 2 different, OCAR8 and SCO3. They are 2 commonly used ovarian cancer models, ovarian cancer cells and 2 different types के. उनके अंदर टेस्ट किया, identified कि डोज ये पैक्लीटैक्सल का डोज है, and this is only combination study. So you look at this, when the drug which is in the market, जो कि टैक्सॉल जो यूज़ होता है, when you give this drug, टाइम के साथ में जो है वो उस पर ज़्यादा असर नहीं करता है, वो बढ़ता रहता है. रेजिस्टेंस के कारण ट्रीटमेंट का असर नहीं होता, जो है द कैंसर जो वॉल्यूम है उसका बढ़ता जा रहा है. व्हेन यू गिव द कॉम्बिनेशन विद द न्यू ड्रग, इट सप्रेसेस, उसको नीचे ले आता है. दैट गिव्स अ प्रूफ कि ठीक है तुम्हारा आपका जो है ड्रग काम कर रहा है. नाउ दिस इज अगेन इन xenograft model. Remember, mice, and then I will come to patient-derived xenograft model. You take the patient sample, implant inside the mice, let it grow. So it's a human patient sample which is inside the mice. And then you treat that to see now it is working in the patient tissue cells or not. So we did that. And then another thing is to toxicity and to metastasis. Now this is one control जो ट्यूमरॉइड्स स्पेरॉइड्स जो बनते हैं वो उसका वॉल्यूम कितना है, कितने बनते हैं, दैट्स अनदर वे ऑफ इंडिकेशन ऑफ कैलकुलेटिंग वेदर योर ड्रग इज वर्किंग ऑन आर्ट्स. सो एज यू सी हियर कंट्रोल जिसमें कुछ भी नहीं दिया गया, पैक्लीटेक्सेल जिसमें दिया गया फिर भी उसमें थोड़े भी बच जाते हैं, ड्रग अलोन उसमें थोड़े भी काफ़ी वो साफ कर देता है, थोड़े बच जाते हैं. बट व्हेन यू गिव द कॉम्बिनेशन ऑफ दिस पैक्लीटेक्सेल एंड SU56 एज यू सी हियर सिग्निफिकेंटली चेंज फॉर यहाँ से लेकर उसको सप्रेस करके यहाँ पहुँचा दिया. Right, and if you look at this combination, control, our drug alone, paclitaxel alone, but when you give the combination, वो उसको एकदम suppress करता है, जो tumoroids जो हैं, tumor size एकदम से suppress करते हैं, which means it shows that in combination of the new drug, your paclitaxel, which is the drug used in the clinic, combination में lot more effective होता है, and we have shown that it happens because the drug, so in a very simplified way, what I've been telling you is कि जो टैक्सॉल जो है उसका रेजिस्टेंस होता है बिकॉज ऑफ वाइबॉक्स प्रोटीन 1 YBX1 प्रोटीन. इफ यू टेक ए ड्रग जो कल्प्रिट है, जो गुनहगार प्रोटीन है, जो प्रोटीन विच इज़ द ड्राइवर, आप उसको सप्रेस कर देंगे तो जो आप कमर्शियल ड्रग ले रहे हैं उसकी एक्टिविटी बढ़ जाएगी, फिर से वो काम करना शुरू कर देगा. करेक्ट, एंड दैट्स व्हाट यू सी हियर कि when you combine, why combination is working, because when you give the new drug, the drug that my lab has been working on, if you give that drug, उसने वाइबॉक्स प्रोटीन 1 उसको दबा दिया, अब अगर आप पैक्लीटेक्सेल देते वो काम करता है. तो जो ट्यूमर वॉल्यूम है वो ज़्यादा से लेके एकदम नियरली यहाँ से देखिए, दिस इज विदाउट ट्रीटमेंट, विद कॉम्बिनेशन ट्रीटमेंट इट इज सप्रेस. सो दैट गिव्स यू एज ए साइंटिस्ट मोर कॉन्फिडेंस कि आप ठीक दिशा में जा रहे हैं. यू आर, and then we expanded those combination studies again, as you see here, as you see again imaging, we use the imaging to follow, to study how much it's affecting. So you see control, single drug, when you do the combination, as you see the volume goes down. Okay, so we tested the same thing. There are various different types जिसके अंदर जो है ये Y-box protein का expression. This is TCGA data, the cancer genome atlas. It's called TCGA, Cancer Genome Atlas. That data shows, which is, it keeps on, the NCI or NIH जो है, they collect information from patient information, they keep on updating and they keep on analyzing mRNA level के ऊपर. So then we looked at which are the other ways, and then we identified again TNBC के अंदर है, which I started to tell you. We looked at TNBC. Same thing, we studied in vitro study, again to look for what are the drivers, which proteins go up and down before and after treatment. Again, Y-box protein turned out to be one of the driver. And then, as I was telling you, showing the structure of beta tubulin, how many GTPases, how many kinases, it's a complex soup. To identify कि Y-box protein actually कहाँ पे काम करता है, there are many mRNAs, there are many mRNAs there, right? So we do that, and then one of my postdoc spent nearly year and a half एक RNA को पढ़ने के लिए, एक mRNA को पढ़ने के लिए. And then fortunately for us, when we were working with this, so one of the Nobel laureate actually had in his Nobel lecture mentioned about this protein, and then we worked on that, and we finalized, and we sent him compound, and he's looking at the structure, at the mechanism of that. So anyway, we narrow it down to one RNA, because RNA is small RNA, large RNA, कौन से RNA पे actually Y-box protein का असर है. We did a lot of these studies to narrow it down to what could be the mechanism. So I guess in the interest of time, these are the people who worked. I'm just standing here and talking. And with that, so I hope you all get the point. And people were asking me yesterday about contact में क्या प्रोफेसर गुरजसप्रीत सिंह से तो मिल ही सकते हैं, मैं उन्हें तकलीफ ना दें इसलिए मैंने यहाँ इसमें दिल लगा दिया है. Feel free to reach out to, if you have any question. Okay, so I hope in past few days, as I was trying to tell you, and now this is experimental, therapeutics is experimental. I hope as I was able to show you how this whole enterprise works. It's a multidisciplinary effort. A lot of expertise, lot of resources, lot of मशक्क़त करनी पड़ती है